Hello, everyone. This is Gianna Chovy with MultiCASE. I'd like to thank everyone for joining the call today. I have with me Rustam Sayakov, President of MultiCASE, and Suman Chakravarti, Vice President and Chief Scientific Officer of MultiCASE. Before Suman gets started with his presentation today, Rustam will take a small amount of time to introduce Suman. So I will go ahead and pass the mic to Rustam. You should be able to see his screen. Go ahead.
Thank you, Gianna. Hello, everyone. It's hard to say what time of the day it is. For us, it's very early morning. For you, it's probably a good day. For some of them, maybe even good evening. I have a great pleasure to present to you today our today's speakers, Dr. Suman Chakravarti, who is holding two positions at MultiCASE. He is Vice President and Chief Scientific Officer. Suman was born in India, and he received his full education at Devi Ahilya University. He passed through all stages, starting from bachelor in pharmacy all the way to PhD in medicinal and pharmaceutical chemistry. In MultiCASE, Suman is performing multiple roles, and he is mostly generating ideas and converting them to actual products, plus he's overseeing the research. He is principal author of Case Ultra and Meta Ultra software, and his area of expertise is computational chemistry, QSAR, cheminformatics, and programming, obviously. He published currently above 20 publications in peer-reviewed journals. I'm sure that this number will triplicate by the time of his retirement, maybe even more, and he holds 2 patents. Suman can speak in multiple languages. Today he chose to speak English, and I welcome Suman in his presentation. Please listen, you will enjoy it. Thank you very much.
Hello, everybody, and thanks, Rustam, for your kind introduction. I welcome everybody who joined this webinar, and good morning and good afternoon, depending on which country you are joining from. And I thank you, all of you, for joining this webinar in this difficult time, and I wish very well for everybody. And I also thank Gianna for arranging this webinar. I understand it's a very difficult venture to design such a webinar in such a short period of time and do everything from a logistic point of view. So with that, I want to start the presentation. And today, I thought about talking about application of expert knowledge in the view of ICH M7 impurity classification. When I was writing these slides, and before also, whenever I presented such a presentation and topic, I always realized that it's kind of really difficult to present the application of expert knowledge in terms of impurity classification, because every impurity is different, and every response for analysis of a particular impurity is also different. But what I thought that it will be good to find out the basic fundamental principles of such a classification in terms of ICH M7, because there are certain common things that can be transformed or transported from one problem to another. There are certain basic things that are all same, and if I can highlight those, maybe that will help you in reviewing such impurities, and that will also help you in gathering experience. And I also assume, and I'm sure that in the audience there are lots of people who already have done such a job for a long time, and for them hopefully there will be something interesting. So with that, let's start, and let's first check out what is the ICHM guidance. I'm sure majority of you already know about it. In a nutshell, it is assessment and control of DNA reactive or mutagenic impurities in pharmaceuticals to limit their risk of carcinogenicity. So that is the basic objective of ICH M7 guidance, and it applies to impurities in drug and mainly pharmaceutical compounds, and during their clinical development and when you are submitting application for their marketing. So basic objective and for ICH M7 guidance is that when you are producing drugs, there are reactive reagents that are used mainly to increase the yields of substances, final substance. So those reactive substances can also react with DNA if they are present in the final compound. So that is the assumption behind all ICH M7 guidance. And it also applies to impurities that are already approved drug substances and products and can be used for excipients that are used the first time and which are chemically synthesized. So as you can see that all the various aspects of the guidance is geared towards synthetic substances because they are the most liable for having such impurities that are DNA reactive. The guidance doesn't apply to drug substances and products that are intended for advanced cancer indications, because as you can imagine that those substances, they themselves are mostly mutagenic and genotoxic. So that's why it doesn't apply to them. It also doesn't apply to drug substances that are itself genotoxic at therapeutic concentrations, as I already mentioned before. And they are not intended for, the guidance is not intended for biological, biotechnological, peptide, radiopharmaceuticals, fermentation, herbal, crude products of animal and plant origin, also oligonucleotides, because they are not produced through synthetic routes. So there are less chances of having reactive impurities in them. So that's why the guidance is not intended for. It is also not intended for existing marketed products, excipients, flavoring agents, colorant, and perfumes. It is also not intended for leachables in drug packaging, but can be used if warranted. So for leachables, as we have heard many times that people actually use, in silico systems to find out their impurities. The ICH M7 guidance is not primarily intended for them, but they can be used if needed. Another thing that I forgot to tell you at the beginning, that is the whole presentation is designed specifically for ICH M7 guidance, but there are certain things in the presentation that can easily be applied to other QSR applications. So I'll be giving examples that are specifically related to ICH M7 and mutagenicity predictions, but you can take out certain principles and you will recognize them easily and can take them and put them into other types of problems that are related to QSAR. Okay, so let's go to the next slide. And this is the basic overview of different types of impurity class according to ICH M7 guidance. And the first three classes, class 1, 2, and 3, which are shown in red color, they are mutagenic impurities. And the last two, 4 and 5, they finally, those impurities are classified as non-mutagenic impurities. And I'm trying to avoid into going into details of each part of these impurity classes, but it is sufficient to say that the first two classes, class 1 and 2, you do not need to have any kind of in silico prediction system for them, but you need to do database search to find out existing experimental data regarding those compounds. For example, class 1 means these are known mutagenic and carcinogens. Class 2 means they are known mutagens with unknown carcinogenic potential. So these are basically related to database search. And the second class, the class number 3, 4, and 5, they would need QSR prediction because there is not data available for their known mutagenicity potential. So you need to predict their mutagenic possibility or any kind of alerts that are present in them. So this is basically related to prediction. So these two classes are fundamental for expert review application because first one is related only to database search, and the second class, which is 3, 4, and 5 impurity class, are related to QSR prediction. So prediction means you do not have enough experimental data or no experimental data, and you need to find out if this is going to be mutagenic or not. So for those, we need to have a QSR prediction. Now, this whole thing can be put into broad risk assessment of impurities, and it contains several different steps, and I'm not expert in those, majority of them actually. So it starts with compiling the list of impurities that might end up in the final product, and that also require expert review on its own. And then the mutagenicity assessment of impurities using in silico methods. This is what we are going to talk about today. And then evaluation of exposure, establishing safe limits, and determination of risk. So these are different parts, and each part warrants expert review. However, in this particular presentation, I'm going to be focusing mostly on the second point, highlighted with red color, to do expert analysis in terms of mutagenicity assessment of that for the impurities that you already know exist, or you envision that they're going to exist in the final product using QSAR and in silico methods. So that is going to be my main focus in this presentation. Now, why we need to have expert review? We can ask that we have a QSAR software. We predicted the impurities potential of being mutagenic, or we have searched in database. Then why do we need? The main problem that actually warrants expert review is that you can have all kinds of problems while using a software. So one of them is that you get conflicting predictions from the two systems. And when I say two systems, that means from a point of view of M7 and QSR analysis, I did not cover it before, but it needs two different methodologies: expert knowledge-based system QSAR and statistical system QSAR. So if you get different predictions from these two systems, then you need expert review. Also, there are other problems associated. You might get predictions that are not conclusive. You get out-of-domain predictions or out-of-domain outcomes. Or majority of the time, we want to find out the validity of the identified alerts in the query chemical. So then also we need to do expert analysis. Sometimes we need to do mechanistic interpretation of the alerts that are present in the chemical, or some cases, which I'll be covering in further slides, we need to incorporate data from analogs or chemical compounds that are actually not present in the QSR system. So in those cases, we have to apply our own understanding of chemistry or toxicology to incorporate that data into final prediction. Also, there are other factors that has to be taken into account, and that include physicochemical properties of the impurity or ADME properties of those compounds in human body. So these factors, they are not taken into account fully by the QSAR system. So anything that is outside of the QSAR model that you are using, it's not able to do a good job with those. So then we have to apply our own understanding of the whole system to get a final decision. Okay, so the highlighted point is the one that we see the most use of expert review in terms of ICT M7 to determine the validity of identified alerts in the context of the query chemical. Now, I'm going to classify the expert review into two different sections, as I have already shown at the second slide, that are, first one is the expert review for class 1 and 2. So these expert review is relatively easy because it is just applies to finding data that is relevant to your chemical, and it involves a simple database search to find out if adequate data is available to prove mutagenicity or carcinogenicity or absence of it. And it includes trying to find Ames test data for the impurities if it is already present, or rodent carcinogenicity or human carcinogenicity potential data for the chemical. So as you can imagine, that if there is an impurity that you come across, which people have already tested in Ames test with fully compliance with GLP conditions and all the regulatory strains and everything, then there is no need of predicting its potential. And those are the class 1 and 2. So in those cases, what you need to do, you need to just find out if this data is adequate or not. And you can also use your in-house database, if you have one, to find out if you have already done an testing on a particular impurity. So any database that you have access to, which is a standard database, fulfills all the criteria, can be used. In case of MultiCASE specifically, we have very nice mutagenicity and carcinogenicity database, and that can be used to search existing data. If the compound doesn't turn up to be one of these in our database, MultiCASE database, you can also search in scientific literature. Many times I just Google it to find out if there is any data available. That doesn't mean that you can cite Google, but at least you can find out pointers from Google to go into specific sources or specific scientific papers to find out more about the chemical. So anything that you have access to can be used. Of course, when you will be submitting this data to the regulatory, you have to document it properly with proper scientific publications, literature, and experimental data references. So that is that this particular database search is comprised of expert review of class 1 and 2. Now, how to search MultiCASE databases? Now, since I don't have access to other vendors' products, I can only give you examples from our own software. And in MultiCASE databases, it is extremely easy to do any kind of database search. What you need to do, you need to just test the compound like any other test, and it will automatically do all the database search that is needed. Anytime you perform a test in MultiCASE, it's a database search first. Then it goes into prediction if nothing is available. And in that respect, we have a tool called Consolidator that provides detailed mutagenicity and carcinogenicity experimental data whenever you test a chemical. Rustam, who spoke at the beginning, he builds this database, and I can tell you that I have seen this is one of the finest databases that is available in the world right now. It contains extremely detailed literature review of compounds, experimental details, conditions, very carefully done and continuously updated. So that is the beginning of your test if you are using our software. And whenever you test a chemical and you get this kind of outcome, known negative or known positive, that indicates that we have experimental data for those compounds. And as the time goes by, our databases becomes more complete, bigger, and with more refined data. So known positive, known negative outcomes means we have experimental data for those compounds. So I'm just showing this example because I want to highlight that for class 1 and class 2, finding the experimental data is the most important part. Now, this is how the experimental data from Ames Test looks like. It includes what kind of strains that has been used to test a particular compound, what kind of metabolic activation has been used, what kind of test method was used, and other experimental details. So this is a simple screenshot from our one of our predictions where we have the experimental data for a particular compound, and this is how the raw references comes out for a particular compound, you know. Now, this is a detailed screenshot of the results of carcinogenicity data if available. So in this case, I have searched NDMA, nitrous compound, which is infamous nitrous compound. And as you can see that we have carcinogenicity data available for this particular compound, in rodents, as well as IARC human carcinogenicity data available. This is not actually a compound that has been tested on humans, but there is an IR group who did expert review of this compound and came to the conclusion that this is probably carcinogenic to humans, which is true. So carcinogenicity data is also important if available. Majority of the time, you'll not find carcinogenicity data because not all the compounds have been tested in humans or rodents. Of course, not in humans, but not also in rodents. But whenever available, it is good to see to those data to find out what is the ultimate effect of any mutagenic compound into living organisms. So in this case, rodents mostly. Now, what is adequate experimental data? Sometimes you have a compound for which some data is available, but not all the data. So that's also not sufficient for class 1 and 2. If you are citing a compound to be not class 1 and 2, you should have adequate data. That means it should have been tested in at least 5 strains of bacteria. That includes Salmonella and E. coli strains. As you can see, they cover different mechanisms of mutagenicity. That's why these 2 different types of bacteria are used, E. coli and Salmonella. And if a compound is not tested in all these different strains, different types of categories of strains, the experimental data is considered to be inadequate. Also, in addition to that, they have to be tested in the presence and absence of metabolic activation, because some compounds can become mutagenic after they undergo metabolism. So that is why if you do not add metabolic activation into the Ames system, Ames Test assay system, it is incomplete. Also, other specifications are there. Say, for example, what kind of concentration has been used, and if there is carcinogenicity data available, that has to be adequate also. So just having experimental data is not enough. It has to be adequate. Now, in order to determine if a compound is inactive or negative in mutagenicity, you need to have evidence that it has been tested in Ames test and it is negative in all the 5 strains that I just showed, with and without metabolic activation. So it's for negative compounds, the requirement is a little bit more stringent as compared to deciding if something is positive or not. So in negative, it has to be negative in all the 5 strains with and without metabolic activation. If that is available to you, and if the Ames test has been done with proper specifications, you can say that this is definitely doesn't belong to 1 and 2 class 1 and 2. But in the case of positive, you just have to show that it was positive in at least one strain with or without metabolic activation. So as you can see, that to prove negative is much more harder as compared to prove if something is positive. Positive means mutagenicity. So right now I'm talking only about mutagenicity, not about carcinogenicity. So this is, we should keep in mind, and this actually applies to all the different parts of analysis. Now, let's go to the more interesting classes of impurities, class 3, 4, and 5. And this is fundamentally different from class 1 and 2, because in this case, the expert review need QSR prediction. Would start with QSR prediction using two different systems, statistical and rule-based. And in our case particularly, you can get predictions which belongs to 4 different categories: positive, negative, inconclusive, and out of domain. Now, positive means, and please keep in mind that these are all predictions. That means this compound is not present in our database. So the software is predicting or estimating if this compound is going to be mutagenic or not. So that is why it is called a prediction. So this is a completely new compound that we have not seen before. In our database, it is not present, and the software is going to make a prediction. So positive means the software has identified, or the system model has identified, presence of strong alerts, mutagenicity alerts. These are functional groups that has something to do with being mutagenic. And if it is negative, either there is no alert identified, and it doesn't matter if it is expert system or statistical system. This is same for both. If no alert has been identified, it is automatically considered as negative or not positive, at least. But that doesn't mean that negative means there is no alert always. Sometimes there could be some alert present, which are either weak or has been completely deactivated by some other functional groups that are present. So those deactivating features are also called mitigating features, which actually brings down the potential of toxic groups towards negative. So negative doesn't mean absence of alert always. It can be, but sometimes there could be some alerts present, which are weak alerts. So when we say negative, sometimes actually we still need to do some expert review to find out if those alerts are truly negative or not. Then inconclusive means there could be some alerts present, and not could be, but definitely the alerts present. But those alerts are either moderately strong or some strong alert that has been deactivated towards lower probability due to the presence of mitigating features. And at the last, which is out of domain, means your chemical contains a feature or a functional group that is not present in any of the training set compound that we have. So this is a new compound. It is unfamiliar to the QSR model. So that's why it is out of domain. So out of domain means no tag essentially. And as a expert reviewer, this is one of the most important responsibility you have to find out if this out of domain is negative or not, or if the groups that are responsible to cause out of domain outcomes, are they mutagenic or reactive? So that is one of the tasks of expert analysis. Now, whenever we talk about prediction outcomes, we talk about two systems, two different methodologies, statistical and expert rule based. And as you can imagine, there are all kinds of permutation combination possible when you are making a prediction. Two systems can view same predictions, like negative, negative, or positive, positive, that I categorized as clear outcomes. In these cases, the decision is relatively easy. If both are negative, majority of the time they are negative. If both are positive, majority of the time they are truly positive. So the burden on the expert review reduces when you have this kind of clear outcomes, agreeing outcomes from the two systems. But keep in mind, that doesn't mean that you do not have to do any expert review on them, because sometimes we have seen cases when the two systems are giving positive outcome, but after expert review, we have enough evidence to call it negative due to different types of reasons. Same thing happened on the other side also. We get negative calls from both of the systems, but after expert review, the chemical turned out to be positive. But the occurrence of such contradictory outcome after expert review is fewer when both of these systems agree with each other. You can also get conflicting outcomes. That means the statistical system gives you negative and expert rule base gives you positive, or vice versa. So those are called conflicting outcomes. And you can also have uncertain outcomes, that one or more system methodologies gives you either inconclusive or out of domain. So we can spend a long time to discuss what we should do if we out of domain and inconclusive or negative or any other combination, but it will never take you to a clear decision, because I believe that whenever you have this kind of conflicted outcomes, that's the time when we should talk about checking the alerts involved and performing expert review. So these are the main outcomes scenarios when we need to do structural analogs search to validate alerts and reactivity of unknown fragments. So unknown fragments means out of domain. So these types of conflicting or uncertain outcomes mostly require expert review using structural analogs. So as you can see that we are gradually going into the place when we need to do more and more review of the final outcome. You know, this happens after you have already predicted the chemical using the two methodologies. So I did not highlight the clear outcomes because those are the cases, as I said before, you need fewer number of expert review. So let's talk about structural analogs. As you probably have noticed that at the beginning of the presentation, I said that some of the features of this presentation you can actually use into other types of QSAR outside of ICH M7, and this is one of them. This can well be used for other types of QSAR problems in case of, say, skin sensitization or say acute toxicity. But today we're talking about ICH M7 and mutagenicity. So let's discuss what are the fundamental need of searching for structural analogs during expert analysis and how it actually helps you. So analog search is done in Case Ultra all the time, and it is done automatically whenever you make a prediction using consolidated tool in Case Ultra. The main objective of doing structural analog is to find out if the alert is valid or not, or if the out of domain is truly out of domain or not. And usually the structural analog search is done in combination with mechanistic knowledge. That means you are trying to find out what is the meaning of this alert in my query chemical, and the analogs helps you to understand the outcome of an expert, not expert, but an alert, in which way it can either cause mutagenicity or is it safe. So mechanistic knowledge in combination with structural analog is quite important. It also, the analog search also adds confidence to the prediction. It can also give you evidence to dismiss an alert or refute an alert. And it can also give you good evidence to fill out of domain data gaps. So the objective of analog search is very, very important, and it includes fill out the data gaps that is present in out of domain outcomes, or you want to find out if my alert is valid or not, or you want to check if my query chemical is really the alerts identified in my query chemical, are they really relevant or not. So in those cases, analog search is extremely useful. And majority of the time, since we are talking about DNA reactive features of chemicals, usually the similarity of analogs that are based on local similarity around the alert is most relevant, as I'll show you later on. Sometimes we can also do a substructural search and global similarity. So there is no hard and fast rule in similarity or analog search. It is done with an objective to find out the meaning of an alert. So you want to find out my alert is truly can cause mutagenicity in the context of my query chemical, and depending on that objective, you have to find analogs that can satisfy that criteria. Of course, you cannot cherry pick, but if you pay attention to certain basic fundamental principles, you'll be able to know or actually you will be able to see there is a regular scientific procedure involved in doing analog search. So here is an example that I have taken from Dr. Naomi Krulok from Cedar FDA, where she actually showed very succinctly how an analog search is helpful to find out the reactivity or the mutagenicity of a particular functional group present in a query chemical. So in this case, we have a chemical that contains an amino group, aromatic amine. So usually aromatic amines are considered to be mutagenic. Now in this case, we have found few chemicals, in this case 5 chemicals that are similar to our query chemical, and they contain the same group. Now as you can see that the first 4 analogs, they are highly similar, at least they look similar, they have the same type of functional groups involved, and they are all negative. And now I have written AES, which is actually different from a classical similarity index. AES means alert environment similarity, where we have calculated the similarity based on the functional group that are present around the alert, close vicinity alert. On that basis, the software gave us these 4 analogs that are all negative. So we can say with confidence that in this particular query chemical, the aromatic amino group seems to be non-mutagenic. I mean, it has the potential of mutagenicity, but the mutagenicity is not expressed in this particular chemical due to the presence of other groups around it. So this is a conclusive evidence that this chemical is non-mutagenic. Now, there is one interesting analog at the bottom where it is positive. But as you can see that I have highlighted the part, this anthraquinone part with purple color, and that is another known mutagenic alert, and that is not present in our query chemical. So that is this alert, this analog can be dismissed. I mean, you can exclude this analog from your analysis because it contains a concurrent reactive group that is absent in our query chemical. So this kind of analysis or this kind of alert finding is automatically done in Case Ultra. You don't have to actually do it yourself. You don't have to find such analogs that are positive and contain different groups and you have to discard them. You do not have to do that. It is usually done automatically in Case Ultra. But it shows you very clearly how a basic analog search is done. Now let's go through some case studies really fast, and I have actually gathered really simple case studies. And as you can see that in this case, there are conflicting outcomes coming from the expert rule-based system and statistical system. One gives positive, one gives negative. Now what to do? So in this particular case, we got multiple negative logs identified for the query compound, and they are negative, and one of them is very close. So this is the one negative compound. As you can see, that the arrangement of groups around the alert is pretty similar to both the analog and in the query chemical, and the analog is negative. And the expert rule also contains, expert rule based system also showed a deactivating feature that actually is capable to inhibit mutagenicity of aromatic nitro compound. So in the basis of these 2 evidence, we conclude that this is a class 5 and negative compound. So this is a very simple case, and I can guarantee you that when the cases that you'll encounter in your real life is not going to be as simple as this. But just to give you some indication of how an expert review is done. In this case, on the basis of analogs and on the basis of a deactivity feature, we are concluding that there is good evidence that this chemical is negative. Now let's go to the case study 2. Again, two different outcomes from contradictory outcomes from the two systems, expert rule based and statistical. And in this case, the alert is identified from both the models, expert rule and statistical. There are multiple similar positive analogs were identified, and that's why we concluded it as class 3. So let's see the analogs that has been identified for this particular query chemical in the next slide. So as you can see, we have identified 5 analogs. They contain the same alert and they are all positive. So it's a clear indication that this chemical is positive based on even after we got contradictory statements from the two systems. Now, one thing people ask us always that how much similarity cutoff is required. You know, personally, from my own studies that I have done, there is no hard and fast rules. It depends on what kind of functionality is present in your analog. So from my point of view, similarity is just an excuse to get proper analogs. And once the analogs are there which satisfy the question that you are asking to determine the outcome of your query chemical, that analog is fine. Doesn't matter what is the you know, the similarity index. Of course, if there is a chemical that has similarity of 0.1, and if there is a chemical that has a similarity index of 0.7, you would trust the 0.7 more compared to 0.1. But sometimes the first similarity is 0.2, you know, because it has other types of functional groups around it. But if it satisfies the question that you are asking, it still can be used. Now this is a case study that actually contains an out-of-domain outcome from the statistical system and a negative from expert rule-based system. So the function group that is causing the out-of-domain is highlighted here, and that part is unknown. It is not present in any of the training set chemicals. That's why the statistical system is calling it out-of-domain. But we found a chemical analog that contains the same fragment, that is a negative one. So this is an analog that contains the same type of fragment, a methyl group, then nitrogen, and a sp1, you know, sp1 hybridized carbon, triple bonded carbon, and it is a negative compound. In addition, we have found multiple similar analogs based on global similarity. They are all negative. So on the basis of that, we conclude that this is a class 5 compound. And here are the analogs that we identified for this compound. So as you can see, these are all similar compounds to the query chemical, and they are all negative. So you see, everything depends on the type of question we are asking. First, the question we ask is the unknown fragment is unreactive. Based on the analog, the answer is yes, it is unreactive. It doesn't seem to be causing any kind of mutagenic. Once we are satisfied with that, we can have more evidence based on the whole structural similarity, finding out if there are enough evidence that the whole molecule is inactive or not. And yes, we have 5 very similar compounds that are negative. So on the basis, we call it negative. Now here is an interesting study. Here, the alert that is present in this impurity, and this is an impurity of acetazolamide, and this alert is present in the API, and the expert rule based and statistical based system both are calling it positive. So what do you do? The API contains the same alert. Now, based on the ICD-7 guidance, we are compelled to say that, okay, since the API also contains the same alert, it is negative. But the problem is, in that case, here is the API. You can see that the environment around the alert is very different in the impurity and in the API. So what should we do? We could not find any analogs that actually support this outcome that this particular acetamido group is inactive. So what should you do? So I found one way of doing it, and I don't know if that is right or not, but at least that seems to be a reasonable way to go. That is, this acetamido group usually cause mutagenicity based on its metabolism to aromatic amine. So can we convert it to aromatic amine and then find out if it is if we can make a decision? So here is the metabolite, and I have used our metabolism system to confirm that indeed acetamido group can be hydrolyzed to amines. Of course, we also know, and also literature, it tells me that acetamido groups usually converts to amines. So on that basis, I found this compound, and this compound contains a deactivating feature, which is 2-amino-1,3,4-thiazole. So when amine group is attached to this kind of heteroaromatic system, usually they are not. So on that basis, we can conclude that this is a class 5 impurity. I still have some doubt, but this is one of the evidence that I found, that the aromatic amine, when combined with 1,3,4-thiazole, is a known mitigating feature or deactivating feature, and this is a valid alert in our system, GT expert system. So on that basis, I concluded that this is a class 5 impurity. Now, API requirements. This is one important thing regarding expert review comes out. The basic thing to remember is that API should be experimentally Ames negative compound. You have to have adequate experimental data for the API before you start assessing its impurities using ICTMs and guidance. In case ultra, we always say test the API with the impurity, but the test for the API is not to predict its mutagenicity, but to check if it contains alerts that are present or shared with the impurities or not. So that is the only reason why we tell you to predict the mutagenicity of API, not to actually predict it, but just to find out alerts. Now, case ultra always assumes that an API is negative as soon as you tell it that this particular compound is an API, unless it is present as a known positive in our database. It always assumes that an API is negative. But the whole crux of the matter is that the API should be experimentally Ames negative to comply with ICTMs guidance. Because as you can imagine, if the API is positive, there is no point in finding out if the impurities are going to be negative or not. At least in majority of the cases. In certain cases, you need to find out if the impurities are more mutagenic than the API or not. But I'm talking about in general terms, majority of the cases, you need to have totally adequate experimentally negative data for the API. Now, one important thing after the expert review is to prepare a report that you are going to submit to the authorities. Now, usually we don't talk about that that much, but it's quite important actually. If you do not submit your report properly, you can easily assume, and you can be 100% sure that the person who would be reviewing your results in the FDA or other regulatory agencies, he's a human being, so he's going to read it. So we'll have to prepare the report in such a way that it's easily readable, it's adequate, properly documented, and clearly shows the evidence, mainly in cases where the predictions and the expert review are going to different conclusions. So let me show you the basic characteristics of a good QSR report. So these are some of the deficiencies of report that we hear from the regulatory agencies, that reports only containing one methodology. In the case of ICTMs, both of the methodology results should be reported, statistical as well as expert rule based. Sometimes the reports only contain some read across or visual inspection outcomes. It's not adequate. The overall conclusions, they are different from the predictions, and there is no explanations provided. And the outer domains outcomes has not been handled properly. please remember that in the case of FDA, at least as far as we know, they treat outer domains as no test. So they have to be properly explained, and inadequate documentation, incomplete. So these are the basic faults, and I'm sure none of you do this kind of faults, but this is just in case to keep in mind, and particularly to highlight what are the good features of a particular report. That's why I'm showing these deficiencies. Now, let's see. Let's prepare a sample report. So the report should start with materials and method, and that should contain what kind of software that has been used, what is the version number, what is the methodology that has been used. So here is a sample section for this particular area of the report from our software. So for what kind of case ultra version this, and the manufacturer of this or vendor of this software, which is MultiCASE. And the methodology that has been used is statistical and expert rule-based system. These are the models with the version number, and these models fulfill OECD validation principles, and the QMRF documents are attached with the addendum. So we always supply you the QMRF documents that actually discuss and describes the requirements or the validations of every model that you have. So these are official documents which are always with your models. So that's the first page of the report. Of course, you have to add other things like which is the organization which is submitting the report, dates and everything. But I'm just giving you the main content. Now, next part should contain your classification that you have arrived at based on your expert judgment, and individual model prediction that are coming from the individual models. And here is a sample. We have used the same example. Overall classification is class 5, which is alerting structure with sufficient data to demonstrate lack of mutagenicity. And in the table at the bottom, it shows you the individual model outcomes from the statistical and the rule-based system. Both are giving it negative, but the statistical system contains 2 alerts, 2 mitigating features. The expert rule-based system also contains a mitigating feature, which is aromatic amines hindered by ortho group. Let's go to the next section, where this is the main part where we will describe the analysis, expert analysis, mechanistic explanation of the alerts. What is the reason behind the final decision if it is conflicting with the model prediction? And if there is an out of domain, what is explanation of out of domains? So in our case, it's pretty easy. Here is the compound with the alerts highlighted, and here are the comments that I have written specifically to tell why these alerts that are present in this compound, they are not causing mutagenicity. So I have explained. So it's negative outcome from both the methodologies. The aromatic amine and the biphenyl ketones are not relevant in this case based on structural analogs, and particularly the substitution at both the sides of the aromatic amine usually suppresses the mutagenicity of aromatic amines, and we have identified a deactivating fragment or deactivating alert from the expert rule-based system. So this is a very simple expert review that is present in a report, in a report that you have submitted to the agency, that you can submit to an agency. And let's see how we will be supporting this report using the analogs, how we can show the analogs. So in the next system, I'm showing the analogs to support or refute a particular alert and to support any deactivating features. So here is the analogs for the example that I've shown in the previous page. And as you can see that these analogs are shown clearly with the similarity with the compound, query compound. What is the Ames outcome? The negative word that I've written in every compound is actually the Ames experimental outcome. So it's a sample report for the analogs. You can also tabulate if you have than one impurity. Suppose you have 5 impurities. You can actually give a full table describing each impurity and highlight the main expert review points. And I have taken this table directly from slides of Dr. Naomi Krulak, who presented these slides a few days back with us. And she has shown this very nice table that really captures the way the compounds impurity should be presented. Each impurity structure is shown, the alerts are highlighted, the individual model predictions are given, and the final class is also given with the explanation at the end column. So this is a nice way of giving reporting multiple impurities of a particular API. Finally, there should be an appendix to show experimental data. And in this case, I have shown the experimental data for the analogs, each analog that I have shown before. And in this case, I have shown for 2, but you should give a a tabular representation of the experimental data supporting every analog with the references. And finally, the appendix should also contain the software output file. So this is the part where we'll be adding the raw file that you are getting from the QSR software that contains the individual outcome from the statistical model, expert rule-based model, and consolidated output in our case. So the basic objective of a report is that you need to really clearly show the supporting evidence, all the relevant outcomes in a very concise, clear, and well-formatted way. A poorly formatted report also obscures the points, and it can cause confusion and potentially problems. So it is a very important thing to present your findings in a nice way. At least that's what I believe. We are coming to the end of my presentation, and in summary, I can say that QSR prediction of DNA reactivity of impurities combined with expert analysis actually increases the confidence and robustness of the predictions. Some of the, actually not some, but a lot of expert review only requires database search for the class 1 and class 2 compounds, where you do not have to go into any kind of prediction details, just find out if this thing has already been analyzed or experimentally determined at some point or not. Of course, every compound, as I said before, varies. It's different. They are individual compounds, and the expert review also is different for different types of query compounds. But there are some common principles, like analog search, or trying to find kind of out of domain reason of out of domain is. So those are common between different cases. So that we can use to start with. To use a software that is validated properly based on OECD validation principles, which is maintained properly, constantly updated, and with a good support is extremely important. If you get a software that is not maintained, you get it once, and in 10 years it is not updated, it's not actually really helpful because the knowledge changes, we find new compounds, so the software needs to be updated constantly. And of course, support is important, because if you need a question to be answered, if there is a problem that is going on with the software, which happens all the time, the company should be able to support you. So that is very, very critical, at least I assume. I have seen in last 10-15 years that beside the software being really good, the support is extremely important. If a company is not able to provide support when it is needed, and it can come from a simple question or to go to very complex problems, the support is important. And very important is to prepare a adequate, clear, and well-formatted report to submit to the authorities. So with I conclude my presentation. I thank all of you to get some time out of your busy schedules to attend this webinar, and it is my pleasure to give you this presentation. Hopefully you enjoyed it. Thank you very much.